layer of hepatocyte does not mean so much for the PBPK model or in vitro in vivo for the translation into the in vivo human situation. So what we need is to correct the drug amount excreted into the bile pocket. So to achieve that, we have introduced the conditions of calcium in supplementation or removal of calcium to affect the tight junction between the epithelial cells. This was firstly reported by the Kim Brower in the United States. She nicely demonstrated that removal of calcium ions from the culture medium, then hepatocyte cannot keep the tight junction between the endothelial cells. So comparing the drug amount between the in the hepatocyte in the presence of calcium and without calcium, the difference in the amount is actually the amount excreted into the bile. It's a very kind of tricky study, but actually this slide illustrates the time profile of the amount associated with the whole cells in the presence of calcium and in the absence of calcium. So they are all typical, these 5 drugs are some typical drugs which is excreted into the bile. So as you can see, the removal of calcium has actually reduced the cells. That means the amount associated with the difference is what we would like to know. But the problem is that we need to correct the cells for every time point. That is cost too much, particularly when we use the human hepatocyte. The other is that because of the comparison of the cellular accumulation between in the presence and the absence of the calcium ions, so the high uptake in the absence of the calcium may prevent the quantitative analysis because it looks like a high background for the quantification for quantitative analysis. So the other part what we have concern is that how the hepatocyte can express the transporter genes after starting the cultivation. So then we started the collaboration with Phoenix Bios, which produce the human liver transplanted chimeric mice that human hepatocyte can be maintained inside the liver. So that we can prepare the freshly prepared hepatocyte from these chimeric mice, and also we compare the expression with the cryopreserved human hepatocyte. So unfortunately, the expression of some transporter genes rapidly declined after starting the cultures. After 2 days after starting the culture, it drops almost to the zero at the messenger RNA level. But this is the case of OATP1B. But that other transporter shows a different profiles. The starting some expression get recovered after the cultivation. Then we measured the time profiles of the activities. For the OATP1B1, whose messenger RNA rapidly diminished, is that slightly reduce the activity. But for OATP1B3, it decreased quickly like this. But the uptake activity mediated by NTCP maintained to some degree, and for uptake organic cation compound is also maintained to some degree. So it's the maintenance of the transporter expression depend on the transporter molecules. So then we have to care about how long the target transporter is actually maintained during the culture. So we also have some expectation to the emerging cells, iPS, because we can have the chance for gene editing of iPS. This is a result of the collaboration with Dr. Kawabata, who have the skills to develop the iPS to the so-called iPS BMEC. But unfortunately, nowadays, this cell line is not the model of the blood-brain barrier because of lack of some specific marker proteins in these cell lines. But this is just examples of gene editing of the iPS. So we deleted the expression, deleted the genes of some transporter at the iPS level. Then after differentiation, they have a good expression on the apical membrane of the endothelial cells. After deletion of the genes, the activity was dramatically decreased. So this is a very important technique to identify the drug transporters and endothelial cells in human model in future. But once the IPS can have the match rate to the in vivo level. So the other aspect is what we have concerned in the gene editing is that this is a result of the genome-wide association studies, what I showed in the first part of my talk. Then we found some uncharacterized single nucleotide polymorphism that is associated with the endogenous biomarkers like this. So then looking into the details about the human genome, they are not located in the exons, causing the substitution of the amino acid change. But most of the SNPs is located in the intronic part or 3' UTR region and some synonymous codon. So it is not easy to associate with such mutations with the phenotyping. So, but if once we have technologies for gene editing, we can introduce all the SNP into the model cells to reproduce the inter-individual difference caused by uncharacterized SNP in human genomes. So there are such several mutations in the human genomes that cause the changes in the serum endogenous biomarkers. So we have forward the characterization of such uncharacterized SNP using the IPS or other in vitro systems like this. So this is just a summary of my talk. For the preclinical stage and clinical stage, the in vitro model or the animal model is very important for the prediction, IBIBC, or improvement of the clinical data. But we have to consider about how they are precisely translated into the in vivo situation using the reference compound, because the accuracy and the precision are required depending on the purpose of the research and also the development stage of drugs. So what we expect to the MPS, that is very rapidly growing in Japan. So then we are aware the usefulness or attractiveness of the MPS platform in our research. The first one is that obviously that is more physiologically relevant format. So we expect that the cells surrounded in the condition is more close to the in vivo situation that may help the more precise assessment or prediction of pharmacokinetics at the early in the preclinical stage. And also we have the concern about the remote tissue interactions, as Dr. Lantz, is the word correct? So she nicely introduced the clear disease models. We have the studies about the normal tissues in one plate, but we have a very strong curiosity about the interaction between the normal tissue and normal and diseased tissue. Actually, we know that the kidney failure affects the drug metabolism in the liver. So, then we'd like to know the how and why and such remote tissue interaction occurs in the clinical situations. And also that we expect that MPS enables a real-time monitoring of drug concentration and cell response that contributes to the high-throughput screening of the drugs in the drug discovery. And also the recently the growing of the RNA sequence or DNA sequence technologies, the MPS will facilitate the single cell characterized in the metabolism activity and the transport activity that constitutes the liver. Because we have information about the population data in the plate that counts the cell number we use is a million. So, but nowadays we can understand the activity of the metabolites and transport activity in the single cells. So then that helps understanding of the heterogeneity of the cell population in the liver and in normal liver and the diseased liver. So we have very strong interest of the MPS. So it's I'm very happy to have here for the good future collaboration with you, MIMETAS, and other 3D culture biologists. Finally, I'd like to also introduce that expectation for the rapid detection of drugs in the in vitro systems. We have collaborated with Dr. Einaga and Dr. Hibino, who developed a diamond microsensor, which is very small needles. But because of this sensor can detect the redox state of drugs, so then they can allow the real-time monitoring of drug concentration. So this slide illustrates just the concentration profiles of drugs in the blood. The diamond microsensor was implanted in the brain. Then they can monitor the drug concentration in 1 rats. So I think that that MPS is very small platform. So it's not easy to correct the drug solution from the system. So this kind of sensoring technology and also the imaging technology is very important for the real-time monitoring of the pharmacokinetics. Oh, sorry, I didn't explain yet. So this is a summary. We have proposed that empirical approach. So this is very important concept to validate how the model is validated, just based on the published information. So there are lots of the cells and cell lines and lots of the devices in the market. But in terms of the application of drug development, it is very important how the model can predict the in vivo human pharmacokinetics. So correct such information is very important. Actually, we expect the advance of the cell technology, but still they cannot maintain all the metabolic and transport function. Not the ideal situation. So then, depending on purpose, that we have to correct, we have to select good lots of hepatocytes or other cell lines for the prediction. Then IPS will be a very attractive format for genetic modification. Finally, I'd like to acknowledge my collaborators. My research have started in the laboratory of Professor Yuichi Sugiyama, who is a very big giant in the pharmacokinetics of the area of the pharmacokinetics. And also we have collaborated with many researchers in the many pharmaceutical industries and Daiichi Sankyo and Pfizer and so on. And also the technology has been introduced from other laboratories. I'd like to introduce the Deguchi for Tokyo University and Kawabata for NIBIHN for the IPS studies, and Dr. Hibino and Einaga for diamond sensors. Thank you for your attention.
Floor is open for some questions. I can start. So, as you mentioned, the microphysiological systems promise to improve on the in vivo situation or on the in vivo cultures in a few different ways, and that can be by ensuring that certain types of transporters are more highly expressed or expressed more closely to the in vivo situation. But there's also been a lot of efforts going towards improving the longevity of tissues, and of course by really physically changing how these cells are cultured to give you access to different compartments or even enable interaction between different cell types.
Yeah.
So in your experience, which of these different aspects is most inhibiting at this moment? Is it really the presence of transporters or the interaction between cell types?
Oh, it's a very important question, but also it's a very difficult question to answer because we have starting to find a solution now. So actually that the most problem that prevents the translation is that the cells cannot maintain correctly for a long time.
Yes.
So but we expect that MPS can suppress the factors producing in other cells, and also the oxygen and other nutrients for continuously. So that's, we expect that such technological advance will improve the stability of hepatocyte or something to promote the drug screening efficiently.
And are there specific transporters that are the most limiting in research at the moment?
That is the important one is the OATP1B1.
Yes.
Yeah, that is very obviously. But that is the also can be quantified by the other formats of the cell culture. So although the number of drugs is very low, but the canalicular efflux transporter is actually what we would like have the in vitro systems. Yes. So like MRP2, PGP, BCEP, and so on. Actually, that they cannot keep, that the current situation is not very easy for measurement because of the many kinetics reasons and also the biochemical reasons. But the reason why I don't refer to the canalicular efflux transporter is that it's very difficult to correct the bile from the hepatocyte. That is beyond the MPS systems. So I hope there's some connection of the canalicular network with the bile duct epithelial cells by the technology or cell culture technology or engineering is necessary to advance to produce the ideal hepatocyte format.
Yeah. And do you see a lot of value in also connecting up completely different tissues and organs together, or do you think it's for most research you would want to focus on individual tissue types because otherwise your chemistry becomes too complex?
Yeah, it's also carries have several problems. Is that one is the cost because the making the whole body chip may cost very much. So, but maybe cheaper than the clinical study, but beyond our budget in the one laboratory. So that is one difficulty. But hopefully, ideally, it's very good for us to measure the whole body pharmacokinetics in the one chip. It's very excellent because of we can include the various factors, various covariant factors in the body chip to simulate the human PK data. That is very important because that conducting the DDI studies costs very much, and also particularly when the variants of genetic variants is very rare, it is practically quite difficult to examine elucidate the clinical relevance of such SNP. But information of such SNP is very important in the clinical settings, even though that is very rare SNP. So I think that I expect that some MPS open the door to some clinical simulation in the in vitro systems. That is very fine to me. But please tell us somehow discount price.
I think we're getting close to looking at interactions between some tissues.
Yeah,
I think the full body chip is still quite a ways away at this point in time. Are there any other questions from the audience?
So thank you very much for a very nice introduction for the pharmacokinetics study. And I know that you are doing a lot of study in silico simulation modeling and relating to the multi-body organ chip. What do you think about the good point or the point of the organ ownership or in vitro study to in silico study or vice versa?
Because as far as in silico, it's very versatile. That we can easily modify the chemical structure to estimate the PK parameter. So it's a good point and strong point. But on the other hand, we know that the predictability of the in silico approach have many outliers. So then, in a general story, it's okay to conduct some so-called machine learning. But just looking at the one compound, it is very difficult to believe the result of the in silico prediction. And the other point is that MPS strongness of the MPS systems in the PBPK model format is that previously we only focused on one tissue models, liver and kidney. So we have not concern about the interaction between the different tissues. But that was not included in the PBPK model. But now we can evaluate such interaction between the liver and the kidney. So then, then we can include such information in the PBPK model to increase the to make across the PBPK model to more human situation. So then it's very important to have not only developing or focusing on the PBPK model, but also the many formats, to finally integrate each information to the PBPK model for the increased reliability of the computer simulation.
Thank you. So I think with that we can then move on to the next speaker, which is me.